health officials are working to pinpoint the source of the virus. Ground Zero, a market in Wuhan.
The new coronavirus's rapid rise in China in December 2019 got the attention of Duke researchers.
I think it caught us all by surprise just how fast everything went down.
Duke's Human Vaccine Institute quickly shifted gears, helping develop COVID testing and setting up clinical trials for new vaccines.
We're uniquely placed to be able to respond to pandemics.
And it's already working on a new vaccine to help prevent future pandemics.
That's a vaccine that would hopefully have some efficacy against the next coronavirus outbreak.
The speed at which the current vaccines were made have some reluctant to take them.
About a quarter of eligible Americans still haven't gotten their first shot.
It is very frustrating because I just don't understand the arguments against taking the vaccine.
A vaccine that wasn't really made in a matter of months.
The work has actually been going on for many, many years,
giving us all our best shot at ending the pandemic.
Hospitals in Wuhan under-resourced and overwhelmed. People anxious about the mysterious virus that has triggered extraordinary measures across China to contain it.
As the new virus spread across China in late 2019 and early 2020, researchers at Duke University's Human Vaccine Institute were paying attention.
The coronavirus wasn't really a focus of what we were doing here, so we all had to get up to speed very, very quickly. There were a lot of papers that were read and a lot of conversations that were had, but honestly, the moment that we knew it was going to be an issue, we also knew that we were a group that was really poised to do something about it.
If the antibodies are able to bind...
The Institute is a collection of laboratories employing nearly 200 researchers.
We work both on vaccines as well as just basic biology to try to understand how infection occurs and then how the body fights it off. And by understanding how that basic mechanism works, we can then design countermeasures, be it antibodies or vaccines, that can employ those same types of mechanisms.
At first, Duke researchers thought the new virus might be like the first SARS outbreak of 20 years ago. That virus was less transmissible, so easier to isolate. That wasn't the case with this new coronavirus dubbed SARS-CoV-2.
Authorities confirm the virus can be transmitted from human to human through coughing, sneezing, or other personal contact.
And so it quickly became evident that that type of virus was going to be a lot harder to stop. And a vaccine was really going to be necessary early on.
The virus quickly spread across the country, and hospital intensive care units started filling up with patients, including ones here in North Carolina.
There was a huge amount of research that had already been done on coronaviruses because of the SARS and MERS outbreaks that had already happened. So really a lot of the basic research that would have been needed to figure out how do you even make a vaccine against a coronavirus, that had already been done.
The federal government was already working with pharmaceutical companies on vaccines, so the Institute looked ahead. It suspected that the new SARS-CoV-2 virus would mutate.
So what we decided to do was to not compete with what was just starting, but to look ahead to making vaccines that might be used for boosters and might be used against mutants that would mutate away from the current vaccines that were being made.
The Institute collected antibodies from infected people to begin that work. It also helped with clinical trials for the new vaccines and treatments being developed.
We have a clinical trials unit within the Institute that really became instrumental in some of the early trials to test different interventions to see if they were able to stop the COVID-19 disease. And we also had a group here that really kicked into high gear and started doing testing. And so testing became one of the major roles that we served.
Researchers think this trial could lead to an emergency FDA vaccine approval by September. They'll take longer to ramp up production and roll out.
When vaccines did roll out in January 2021, my wife and I got ours as soon as we could.
Have you tested positive for COVID-19 and been instructed to isolate in the last 10 days?
I have not. We were both 65 and had underlying health conditions.
I'm 65, going to be 66 in August. So we're trying to encourage folks to take the shot.
I got my first shot January 15th at the Cumberland County Health Department's drive-through clinic at the Crown Coliseum.
She's giving me the shot right now. Don't even feel the needle at all.
We received our second shots on February 10th.
No lollipops the first time, no lollipops the second time. But hey, it's official. Got the second shot here.
In August, I had 10 people at my house to celebrate my birthday. Everyone said they were vaccinated, but 6 of us got breakthrough infections, including my wife and me.
We took every precaution. The Delta variant does not care.
My wife and 4 others were able to recover at home.
I, unfortunately, did not fare as well.
Now I'm stable because I'm just sitting here. But if I get up and move around, I start having a coughing fits and breathing difficulties.
I spent 9 days in the hospital but recovered thanks to being vaccinated. Researchers say no vaccine is 100% effective, and breakthrough infections like mine should not discourage people from getting them.
All the data is showing that those who have been vaccinated that get infected have a much milder course of the disease and do not end up in the intensive care units or dying from severe infection and the complications of that.
Researchers say people should not be worried by the speed with which the new COVID-19 vaccines were developed and distributed either. They say the infrastructure for clinical trials and manufacturing was already in place.
They didn't cut out any of the steps. They just cut out a lot of the red tape and the delays.
Early research on HIV also helped.
The work has actually been going on for many, many years, and we're fortunate that the technology was there at the time the pandemic struck, so that these new vaccines could be brought to the public quickly.
Haynes remembers his own experience as a child in the age of polio.
We were all very frightened to go anywhere and to be around other children because of the fear of getting polio and becoming paralyzed. As I remember, it was not too different from the last year and a half with regard to restricting our activity back in the 50s.
Then polio vaccines were developed.
And the rates of polio transmission plummeted and society went back to normal. That's a terrific example of just how effective vaccines can be to get back to normalcy and to stop a pandemic in its tracks. That's what we're trying to do now with the coronavirus.
Next, the decades of research behind the current COVID vaccines.
About 25 vaccines to combat the AIDS virus are currently under study.
So now we're into the main hallway.
And a look inside the Institute's super secure lab.
It's an extremely safe and secure facility for working with the live authentic coronavirus.
The World Health Organization estimates up to 100,000 AIDS cases today.
Duke Human Vaccine Institute Director Barton Haynes began studying what was called gay-related immune deficiency disease for the NIH in 1983.
We were very, very concerned at the mortality rate and the rate of spread of this particular virus. We didn't know it was a virus at that time. We didn't know what it was.
The probable cause of AIDS has been found, a variant of a known human cancer virus.
Haynes studied hemophiliacs who were becoming infected with what became known as HIV.
The infection of the hemophiliacs let us know that this was a virus that could infect anyone in the population.
In 1985, Haynes started working on an HIV vaccine. Among his colleagues was the federal government's leading AIDS researcher at the time, Anthony Fauci.
If we're lucky and we do develop the vaccine, it'll be well into the mid-90s that we have one available.
Haynes says different research grants were going to different researchers all over the world.
We were stuck. We had about 50 different vaccine candidates. Many of them were similar because they were not being compared with each other, and none that, frankly, I or anyone else was really excited about.
Haynes collaborated with the federal government and the Bill and Melinda Gates Foundation to do business in a different way.
And the new business a different way was to fund groups of individuals who would compare the different vaccine candidates and agree to work together.
In 2005, the federal government granted the Institute $300 million to develop an AIDS vaccine.
So what changed in 2005 was that the field began the process of coming together and collaborating in larger and larger groups.
HIV is a more challenging virus than SARS-CoV-2.
It's one of the most rapidly evolving life forms in the virus world. When it inserts into our own genetic material, it cloaks itself, it hides from the immune system.
And our body doesn't naturally make the antibodies needed to fight it.
So what we're having to do with HIV, and what we spent the last 17 years learning, is how do you engineer the human immune system so that we'll safely make those protective antibodies, that when they're made, they don't harm us.
So we know what we need to do, we know what the vaccine's going to have to do, and now it's just a matter of actually putting the plans in place to get that actual vaccine made.
Work on HIV helped lead to the development of what's called mRNA technology for the development of vaccines. It's the same technology used to make the current COVID vaccines.
Typically, vaccines are either virus particles that have been inactivated so that they're not infectious, or they're protein parts of the virus that your body sees as foreign and makes an immune response and establishes memory.
The mRNA platform uses the coding sequence that tells your body how to make proteins.
We can quickly give that to a person, and you use your human body as actually the manufacturing facility to make the proteins, and then your body sees those proteins as foreign and makes the memory immune response, which gives you the protection.
And the beauty of the mRNA platform is it's one that is a very short timeline going from vaccine candidate development going to manufacturing because of just the nature of the way the mRNA product is made.
The technology can be used to make both vaccines and treatments, and it's flexible.
You can quickly modify what goes into the vaccine so that you can pivot to new variants that come out, or take that same concept and apply it to another pathogen that may come down the road.
The Institute's work on HIV vaccine also meant there was already a clinical trial network in place to test vaccines for COVID-19.
And so they were able to pivot very quickly because they already had people in place. They already had all of the staff that was necessary to do it. They just had to turn their attention to a different problem.
The work that's been going on for the past 20 years or so in HIV vaccine development developed much of the technology that was then taken and frame shifted and used immediately in January of 2020 for the COVID vaccines.
Haynes says that's one reason those vaccines were developed so quickly.
People have been trying to make better vaccines for years for a number of different diseases, and all of those techniques were brought to bear on this problem.
The Institute is currently using antibodies it collected from people infected with SARS-CoV-2 to develop what's called a pan-coronavirus vaccine.
When we say pan-coronavirus, we actually mean many different types of coronaviruses, both in humans and both in animals.
If we can get a pan-coronavirus vaccine, hopefully the goal would be you would not have to worry about variants.
The Institute is working on a similar vaccine for the flu.
So the goal there is hopefully to get a vaccine for influenza that you do not have to take every year. So the virology teams that we put together here, the immunology teams that we put together here to study HIV and other pathogens over the years, were able to rapidly frame shift into flu.
Now, at the heart of the Institute is the Duke Regional Biocontainment Laboratory. This is a highly secured facility. It's where you have researchers working with live viruses to develop vaccines. Duke is giving us a rare look inside.
It allows us to do laboratory and animal studies so that we can help develop the next generation of vaccines and treatments to deal with these global pandemic threats.
So Gilbert, we're going to head into the containment side of the facility now. Right. This is a hallway that has 8 individual biocontainment suites that allow us the ability to have multiple research projects going on at one time with different agents. Most of the rooms right now are dedicated to SARS coronavirus 2.
And having the regional biocontainment facility is sort of a jewel within our total program for doing that, because we do have the ability to bring samples in from outbreak situations, be able to safely and securely work with them here at Duke, and then to use the information we learn from those samples to design the vaccines or the therapeutics that will help us to combat that outbreak.
What do you tell someone who's reluctant to get the vaccine?
Well, it's unfortunate that politics and other opinions are coming into play here. It is a very safe, very effective vaccine, both Moderna, Pfizer, J&J. This is something that everyone needs to take to protect themselves, their family, and the community at large.
This lab is also capable of manufacturing vaccines and treatments.
All within the Vaccine Institute here at Duke, we can go from basic discovery all the way to the first-in-human testing, which is quite unique. Very few institutions have their own pilot manufacturing facility.
Next, what Duke researchers learned from this pandemic that will help them prepare for the next one.
Be prepared constantly.
and what I learned from my own battle with COVID-19.
God is good. So is that doggone vaccine.
To watch this WRAL Special on demand anytime, go to WRAL.com and all of these streaming platforms. You can also join the conversation by going to WRAL on Facebook and Twitter.
Average new infections dropping and hospitalizations declining.
As the pandemic appears to be winding down, researchers at the Duke Human Vaccine Institute are once again looking ahead.
We're preparing for future pandemics because it's inevitable that they're coming.
The Institute is looking at vaccines to fight viruses currently in animals that might someday jump to humans.
And go ahead and make those vaccines and put them on the shelf. Test them in humans in safety trials and have them ready to go when a new pandemic strikes, so we can respond even faster. The other component of being prepared is developing a series of antibodies that you can use as treatments.
The idea is to use antibodies to blunt early infection so it doesn't lead to severe disease.
To have those antibodies on the shelf for a whole panel of viruses so that we'll be ready.
Yeah, let's see how this one looks like.
Dealing with this pandemic has been a learning experience for the Institute's researchers.
And so we've been able to kind of come up with a plan for the future so that when the next pandemic or the next outbreak that comes around, we'll be prepared to be able to respond as fast as possible.
And researchers here also hope the pandemic has taught people about how scientific research evolves.
We learn things and we figure out what's right and what's wrong, and then we move and we continue that process forever. It never stops. And so a lot of times I think people go, well, why are you changing your answer? Well, that's because I know more now. I gave an answer before based on the best data available. Now I have better data, so I can give you a better answer. And it's important to understand that we know what we know now, and we will know more tomorrow.
The Institute's technology and experienced staff provide a great learning environment for young researchers like Maria Blasi.
Yeah, they look great. That is actually one of the things that really attracted me to come here and stay here at Duke. What vector are you going to make?
And attracting researchers like Blasi is critical to the Institute's future.
I think there is a lot of investment from everyone in this institute on training the next generation of scientists. So being able to provide like young investigators with technologies and resources to be able to foster their career is crucial, I think, for any institution. And I think we do have all of that here at the Vaccine Institute.
The pandemic was not only a learning experience for the Duke Human Vaccine Institute, it was also a learning experience for me. I covered COVID-19 as a reporter and battled it as a patient here at Cape Fear Valley Medical Center.
I hadn't eaten in 3 days. I've lost 15 pounds, and I'm living what we've been reporting for the past 18 months, and it gives me a different perspective in reporting the story going forward.
I was diagnosed with bacterial pneumonia. No way Tylenol was going to take care of that at the house. They took me in and gave me antibiotics, gave me medicine for my nauseous feeling. I'm on an isolated COVID ward here at the hospital. I saw the patients up and down the hall in the ICU. It is overwhelming to see all the people who are here fighting for their lives. I'm the only one that had shots. I'm the only one without oxygen or a ventilator. But thank God I had the vaccines to keep me fighting at the level that I'm fighting now in comparison to my colleagues. That's what I was looking forward to.
After 9 days in the hospital, I went home to continue my recovery.
Wow. Fresh air. Fresh air. Unbelievable.
One doctor told me that with my age and diabetes, this day may have never come without me being vaccinated. I shared my story on social media and on the news in hopes of inspiring others to get the vaccine.
Gilbert is back on the job today, the first day since recovering from COVID.
The experience gave me a much broader understanding of COVID-19 and its impacts. I now know firsthand what researchers have been telling us all along, that vaccines are our best shot at defeating pandemics.
Love y'all.